again, myelin is fat. Okay? So do that experiment when you maybe volunteer in the newborn ward. You see in the nursery, the newborn babies, you pinch them. There's a long delay between your pinch and their cry because their neurons are all unmyelinated in the beginning. And then maybe, after they start breastfeeding, the breast milk has fat. So over time, you pinch them again, you see the delay is getting shorter and shorter and shorter. And then when they grow, you just go like that, they start crying. They look at you and they start crying right away. Okay.
I'm sorry, it's just your smile when you say that.
So the amount of myelination, of course, it affects the more myelinated the neuron, the faster will be the propagation of action potential. Of course, if it's wider in diameter, it will propagate action potential faster. Just like if you want heavy duty stuff, don't you buy the thicker one, right? The thicker plug, right, for heavy duty. It's the same principle. And temperature. If you ice down back then they didn't have anesthesia chemicals for anesthesia. What did they do? They ice stuff. They ice the part of your skin. Have you ever done that when Baba doing he's way older than that, not just me. But didn't they do that? What do you call it? Opening. Ear piercing. Don't they sometimes put ice and then they just pierce? Yeah, you can do that. So lower temperature will slow down the propagation of action potential. Okay? So as far as the synapses are concerned, don't forget that a synapse will always involve an axon of one neuron. And if it's communicating with another neuron, then it will involve the dendrite and cell body of the other neuron. Or if it's another cell, the cell membrane of like the muscle cell. But an axon always has to have an axon of one neuron. Okay? Did I say a synapse? A synapse will always have an axon of one neuron. So there are two general types of synapses. The one between neurons is called chemical synapse, and I don't want you to worry about this axon. Look at the synapse. Always involves an axon. There's always an axon involved with a synapse. But there's another type of synapse called electrical synapse, and this is the synapse that you find in your heart. You're familiar with the pacemaker of the heart? When you start doing EKG, that's why we have to study EKG, because the heart conducts electrical potential. But there's no space when the heart, as far as the heart is concerned, there is no space between one heart muscle cell and the next. You remember the intercalated disc, right? That's the synapse. That's called an electrical synapse. It's very fast. It's faster compared to this one, because this one has a space. That one doesn't. Vanessa?
So does that mean there's synapse in the heart?
Yeah. The heart has its own nervous network, and we call it synapse. That's how they talk to each other. You're going to study that when you get to the cardiovascular system. So you see, there's always a space, chemical synapse, whereas with an electrical synapse, there is no space. They're united. Okay? And this is what we talked about last, the excitatory and inhibitory postsynaptic potential. Do you have any question on those two terms? Excitatory just means what? You generated what? Action potential, right? When you say an inhibitory postsynaptic, an excitatory postsynaptic potential was generated, that's just a fancy term for action potential was generated. When you say an IPSP was generated, that means there was no action potential. for the next neuron. That's all. Okay? So as long as neurotransmitters are there, that neuron will keep on stimulating the postsynaptic cell. So there must be ways to get rid of these neurotransmitters, just like in your muscle, remember? So they diffuse out, or you see how calcium is very important, see? There's reuptake, calcium has to push them back inside, or enzymes can break them down, like acetylcholinesterase. But the point is, we do not leave the neurotransmitters in the synapse for indefinite time. Once it's done its job of relaying the message, they go back in. It's just like your words, right? Once you're done talking, you close your mouth. Same here. Done talking, bring the words back in there. Or get rid of them. Diffuse out or break them down. But what's the key? What do you need for these neurotransmitters to be able to go back in the axon? It's calcium. You need calcium. Okay? So without calcium, the neurotransmitters have no way of going back inside. They're here. They stay there. And if this was muscle, that muscle will continuously contract. You see it. Which is dangerous, right? If you think about it, you think about your heart, you think about every organ in the body. So this hypocalcemia, even though sometimes you get so tired, and it's usually they show up in the middle of the night, and you're so tired, right? But you have to attend to them. Give them a calcium injection, and it just disappears like that. Okay? Okay. So we're going to skip this one. As far as neurotransmitters are concerned, I just want you to know this one, acetylcholine, because that's the one for your for muscle. I'm sure you know what these are your adrenaline, isn't that? That's your uppers, which means they're excitatory, same with dopamine. And this GABA, this is the only natural inhibitor neurotransmitter. Sometimes if there is imbalance of neurotransmitters, that's when you get the diagnosis of ADD maybe, or depression, or Parkinson's. It's all involving either the breakdown or reuptake of neurotransmitters. And that's why those three mechanisms for breaking down or getting rid of those neurotransmitters, it is so important. Okay? Okay. So now we look at this one. What happens when neurons get damaged? Remember that CNS damage is more than likely permanent. CNS means your brain and spinal cord. If a neuron in the CNS is damaged, it's permanent, more than likely, because of the astrocytes. Remember? Astrocytes, they form scars. If it's a PNS neuron, meaning it's not the brain, not the spinal cord, there's three things that usually, if these three things are present, then more than likely regeneration or repair can happen. Number one, if the cell body of the neuron is intact, where the nucleus is, then of course it can possibly regenerate. Only for PNS, peripheral nervous system. Number two, if the Schwann cells are present and undamaged, remember the Schwann cells, they're the ones that make myelin. If they're present, if they're not damaged, more than likely it will repair. And lastly, the gap should be no less than 3 millimeters. The break should be no less than 3 millimeters. Okay? So, right here. And that's what you call, what will happen is, see, when there's a break, the distal part undergoes a kind of degeneration. It degenerates. And that's called Wallerian degeneration. Wallerian degeneration. So the damaged part will degenerate, and you see there's Schwann cell. As long as there's Schwann cell body was not affected. The break was no more than 3 millimeters. They made. So the injured part will just undergo this kind of degeneration, Wallerian degeneration. And the Schwann cell is the one that is very important, because it's the one that forms what you call a regeneration, like a cast, right? Regeneration to try and coax the severed end to be reconnected. And they're happy again. This is not possible in the CNS. Why? Because there's no Schwann cells in the CNS. Only PNS is where you will find Schwann cells. Okay? Okay. And I think that's all I want to cover for this chapter. The next part is the interesting part, but I want to look at our worksheet. Where was that? Oh, where did I put that? Okay, under the
I found it under files.
It's right here.
Can you write down your name on a sheet of paper and pass for attendance? Attendance, we're gonna discuss the worksheet. The discussion is worth 25 points today. Write down your name. Can you pass the paper?
Absolutely.
Okay, so it will be, you will see it under a nervous worksheet discussion. Okay, 25 points.
Thank you. No, it's just for being here. We gotta turn it.
Yeah.
That's the dural tear, that's in your CNS.
Yeah, so like when we repair those.
Oh, it's just a minute.
We're getting attendance.
So,
And then like tried this to somebody, I was like,
okay, let's do this. What do you think? Now let's try to see. What is the consequence of a reduced intracellular potassium concentration? So look at this one. What did you write? So that means there's less positive inside. That means you're gonna make it more negative or less. So that means you hyperpolarize. So if there's a reduced intracellular potassium, that means you're taking out the positive. You're making it more negative. You're making it more negative. That means hyperpolarized. Okay? Okay, whole bottle. So, by the way, don't do that. Did you know that if you ate a full tablespoon, a full tablespoon, you take yeah. So don't. When we eat, don't we just eat little bit at a time, right? And you better pray that you have healthy kidneys will push it out.
But what about
Okay, so let's look at this one. That's a topic for the renal system. Okay? Let's look at this one. Whole bottle of salt. Of course, don't say the patient is dead. Don't. What effect will this have on action potentials? Let's think. So sodium, whole bottle, what will it do? It will make it depolarize faster, right? So you predict maybe convulsion. It's just firing faster, right? It's gonna fire faster. So maybe convulsions, etc., etc. Okay. Lithium, permeable to plasma, permeable to sodium. Predict the effect of lithium ions in the extracellular fluid if the effect that lithium ions would have on the response of a neuron to stimulus. So it's the same as this one, right? Same story, number 2 and number 3. Same with you depolarize it very quickly, right? Okay. Some smooth muscle has the ability to contract spontaneously, so it contracts without any external stimulation. Propose an explanation for the ability of smooth muscle to contract spontaneously based on what you know about membrane potentials. Assume that an action potential in a smooth muscle cell causes it to contract. So how are you going to explain that? It just constantly generates action. So I look at, it says here, it contracts spontaneously. That word contract spontaneously tells me it does not need a stimulus. Do you agree?
Yes.
So then that tells me that smooth muscle cells, what do you think? Will it have mechanical gates or chemical gates? Will it have mechanical or chemical gates?
No.
No. Correct. It doesn't need, it says it contracts spontaneously.
Right.
That's what, yeah, it's leakage and voltage. Correct. It just constantly fires. That's the same with your heart. That's why sometimes your stomach, you can hear it rumble. That's why you poop when you wake up in the morning. Okay. It keeps contracting. See, did you tell your smooth muscle? No, it just constantly fires. Okay. In fact, that constant firing, you know, and I hope you remember if you take me for Bio 242. That's called migrating motility complex. That's what blesses us. What's in your butt? Isn't that germs? Don't look at my butt, you got a butt too. Don't we have germs there, right? And I'm thinking if there's no contraction, those germs, those sneaky things keep going up. But the contraction, it just keeps pushing it, keep them down there, okay? Number 5, you have 2 nerve fibers, same diameter, but one is myelinated, the other one is unmyelinated. Oh, you know the answer here, right? Which one is most energy efficient?
Myelinated.
Yeah, you got that? Okay, let's go to number 6. Explain the consequence when inhibitory neurotransmitter is released from the presynaptic and a stimulatory neurotransmitter is released from another terminal, both of which synapse with the same neuron. What? So there's 2 neurons.
There's just so much stuff.
One releasing positive and one releasing negative. What will happen? They will negate, so it stays at resting. Right? Don't they stay at resting?
Yes.
Good. Okay. Next, aging, speed of action. Oh, this one, old age. Never mind. So many examples for this one, right? Aging, speed of action, potential propagation, rapid transmission decreases, list possible explanation. What can you write there? So many possible explanations. What did you write there?
You like to have lower calcium?
Lower, yeah, electrolytes, maybe electrolyte imbalance. What else did you? They have osteoporosis. What else did you have? About fat. The fat content may be decreased when you get older, right? Myelination of your neurons, they have decreased. Okay? You may have less enzymes. You need enzymes to break down neurotransmitters, right? So a dog ingests organophosphate poison, and this is responsible for saving the dog's life. This poison binds to and inhibits acetylcholinesterase. So you remember the acetylcholinesterase is the one that degrades acetylcholine. So several substances they could inject are acetylcholine, curare, which blocks the acetylcholine receptors, and potassium. What would you do? What would you inject? What did you choose? So I think, I'm not gonna inject this, right? Because you already have a lot of that. Which means acetylcholine is what? It's a stimulatory neurotransmitter, right? So what do you give them? Yes, it says it blocks the receptors for acetylcholine, right? Okay? Strychnine blocks the receptors. Are you writing your names? Is it passing? Where is it now? Pass. How about the other group right here? Hurry up. Strychnine blocks receptor sites for inhibitory neurotransmitters in the CNS. Explain how strychnine could produce tetanus in skeletal muscles. If it's blocking the inhibitory neurotransmitters prevent contraction, right? So then that means it's not blocking. If it's blocking the inhibitor, what is it not blocking? The excitatory, right? So the excitatory remain unchecked. So they keep stimulating. And that's how it could produce skeletal muscle tetanus.
So what keeps stimulating?
Excitatory is stimulating, right? Okay, so next, Parkinson, result of decreased dopamine in the brain. But these patients can take dopamine in drug form because the drug cannot reach the brain. Oh, you know this one? What is it? Did someone said astrocytes? The astrocytes form blood-brain barrier. How can these same anatomic structures be beneficial to an individual under different circumstances? And we talked about this, right? The blood-brain barrier is beneficial if there is an infection, right? You have any kind of infection in the blood, the bacteria is very difficult for the bacteria to penetrate or pass through the blood-brain barrier. That's the protection part. Okay? Caffeine. How does caffeine affect transmission of nerve impulses? What did you write? What did you write? First of all, is caffeine excitatory or inhibitory?
Excitatory.
Okay, so then if it's excitatory, then you start with the story of, it starts with -17, and you open chemical, caffeine is a chemical, right? You open chemical gates, that's the same story. Okay? Explain how a person with a spinal cord section at the cervical level is still able to perform patellar. Oh, this one we're gonna talk about this when we get to the spinal cord. Even this one. Oh, you know this one already, the answer to this one, right? So these two, we have to reserve for spinal cord chapter. What's the answer to number 14? Enzymatic breakdown, calcium, and diffusion, right? That says, how do you reduce the concentration of neurotransmitters at the synapse? Right? The enzymes break them down, or they diffuse out of the area, or there's reuptake if there's calcium. Reuptake means they go back to the axon where they came out of. This one is what we just talked about, the generation of action potential. Okay, but this one we're going to reserve also because our next chapter will be the brain, the fun part. Are you done writing your names? Or is that paper?